most spectacular aspects of this are these antigen receptors, including antibodies and the antibody equivalent on T cells called the T cell receptor. Um, and this it's some basic structures, but what's really important are the business end. So what's called V here is what binds the antigen. The rest of it is sort of to stabilize that, or in the case of antibodies, to trigger some very specific functions. Have the next slide, please. Next. Oh, yeah, right. So the trick here is these are not just garden variety receptors to bind antigens. In order to bind every possible antigen in the universe, you've got to have a tremendous amount of diversity. And the way antibodies achieve that diversity, the genes, it was discovered by Susuma Tanagawa, among others, in the 70s, is that you have these different pieces of the binding site that come together in random ways. And they have names, but that's not terribly important. It's more that you just have 50 of these and 10 of those, and there are then 500 combinations of those. And then there's some tricks that go on in where they come together that adds diversity. And there even is a mutating mechanism on top of that creates millions of possible receptor binding sites. And why is that? Well, it's because we can't possibly evolve faster than a bug, than a virus. Virus replicating every 30 minutes or hour, humans replicating every 20 years. There's no way you could evolve a mechanism against something like that and any of the bugs out there. So what the immune system does in higher organisms, us mammals generally, and somewhat reduced complexity lower down in the evolutionary tree, is you basically create millions and millions of every possible binding site for an antibody or a T cell receptor, and hope that they connect with whatever bug comes along that may be just mutated in, say, a bat, and is ready to wreak havoc in our world. So next slide, please. Just it gives some numbers on that, and and all you need to do is say look at the bottom line numbers, and you see numbers like 10 to the 11th or 10 to the 16th or something, and that is a very large number, and that comes from all these fancy mechanisms that make take these gene pieces and throw them together to form a fully functional antibody or T cell receptor. And then that gets expressed on one or a few, until a particular virus or other bug comes along that has an antigen that is a component of the virus, like a protein, that triggers, that can bind to that particular flavor of a receptor. And then that process triggers that cell, and only that cell, to divide and make thousands of daughter cells. But because this rearrangement is fixed into the genome, they all are expressing this same T cell receptor or the same antibody. And there's other things, but this has been, this is a huge. This is like the double helix of immunology. Is the rearrangement mechanism producing this incredible diversity that is even exceeds what you express in your own body. Identical twins don't even express exactly the same. They only share actually only about 2% of their antibodies and T cell receptors have identical sequences. So there's so much diversity, and you're just spinning the wheels, the slot machine of antigen gene expression. So many Nobel Prizes have gone on here because it's a big deal. This is unique in biology. No, people have looked for this in the brain, and they've been desperate, actually. Many false reports of something like this happening in the brain. Doesn't happen. No, the brain, you need to wire the brain in kind of fairly reproducible ways. You can't, this randomness is really a adaptation of higher organisms to deal with the threat of microorganisms and the many and how quickly they can mutate. Have the next slide, please. Next. Yep. Oh, there we go. So this is what it looks like at a cellular level for T cell receptors and also structural. So over on the left, what you see is solve structure was actually done by my colleague here, Chris Garcia at Stanford. Well, he was at Scripps at the time. He's now on the faculty at Stanford. It's a beautiful structure. So the blue is the T cell receptor, and it's very antibody-like for those structural aficionados. And what it sees and the binding is not just the antigen, but it's actually the antigen, which is a little peptide bound to the histocompatibility molecule, one of many. And that's a key aspect I'll get into in the next slides. But what I want you to see here is just that this is all happening at the cellular interaction level. It's not like antibodies are like guided missiles that you soup them up and then you go from a cell surface phase, which is the selection phase, to the finished product, which is poured out of plasma cells at a great rate and is meant to home in through tissues and fluids in the body and bind to something like a virus and bind it very tightly and maybe prevent it, like the neutralizing antibodies to the spike protein in coronavirus, either prevent it from getting into the cell by interfering with its apparatus, or targeting the virus for destruction by some of the other mechanisms. But T cells work very differently. They're working in between two cells, and they're particularly looking at peptides and peptides that are bound to these histocompatibility molecules, and that's something unique and different. Have the next slide. Yeah, next. Yeah, there we go. Good. Um, so there are two ways in which peptides are made and seen by T cells, and one is involved in the so-called class 1 MHC pathway, a set of molecules illustrated here. And what happens is an internal protein, say viral proteins that have invaded and are expressed in a invaded cell, but also all the usual proteins that a cell would make for itself without the virus, and all proteins get degraded, and it's as part of our recycling. So you're continually degrading your own proteins from whole molecules that might be 100 amino acids long to fragments that might be 10 amino acids long to amino acids. And then those amino acids are recycled to make new proteins, which are other amino acids. What happens here is then that in the middle of this process, these peptides are picked up by the MHC molecules. And that's a critical thing because you're intervening into this cascade, of destruction and recycling, just where the proteins have been broken down to about 9 or so amino acids in length. And that's when they're bound tightly by these MHC molecules. And it's the same with the other pathway as well. And that's critical because 9 amino acids has just enough information to generally know whether that's belongs there or not. If you captured amino acids or dimers or trimers of amino acids, that'd be useless because those are so common, you couldn't tell friend from foe. But 9 amino acids is kind of a crucial dividing line such that a T cell can know whether that belongs there or not. And it ignores everything that belongs there, everything the cell would make normally. It focuses, of course, on what's different, because what's different might reflect a viral invasion or cancer or something of that sort. So anyway, so the class 1 pathway involves usually internal proteins and made in the cell. There's another pathway called the class 2, and that is responsible for grabbing, internalizing exterior proteins that might be floating around in the blood or whatever. And then that has a whole pathway and another type of MHC molecule that captures it. The other critical thing here to know is that this is a much harder system to fool than an antibody system. That is, antibodies see the surfaces of intact proteins and viruses. And that can be very effective and often is, but it can also be fooled. We know many examples now where the pathogen makes it very difficult for you to make an antibody against the most critical places in the virus, for example. And the most notorious case is the sleeping sickness parasite trypanosome. Basically makes a decoy protein that it coats itself with, but has absolutely no vital use. And then it has 1,000 of these proteins that it could put on its surface. And when the immune response starts to key in on the whatever the first protein is, it switches and puts another thing on the surface. And then that goes on and on. That's why you have this sort of periodic sickness and then relative health and then sickness again, because it's this yin yang, a constant battle with the immune system and the active evasion by just about every successful pathogen is going to have probably more than one strategy to distract and evade the immune system. And so it's tricky. It's not just it's not usually simple. If the pathogen has been around, its whole job is to evade and to baffle and distract the immune system long enough to replicate and to make a lot more of itself. And that's what we're faced with, and that's what you try to work on in vaccinology, is to try to find out what are the really key targets. And we know also from lots of animal studies and human studies that the T cells are a vital part of almost any defense, but they're in the least understood part. And so people by necessity focus on antibodies. But if we knew a lot more about what the T cells are doing, I think we could make more effective vaccines. The upshot of his extensive studies that came out late last year in SARS-CoV-2 infection in rhesus macaques, which are, works. The vaccines have worked well in those. So we have every reason to think that it's a good animal system. And basically, what he finds is that the best thing you could have are neutralizing antibodies in order to not get sick. And he did that by transferring antibodies from one monkey to the other, immunized monkey or infected monkey to another. But the other thing important in this conversation is he found out that if you don't have the absolute best antibody responses, that T cells can save you. That he showed that T cells were really playing an important role in protection if you didn't happen to make the absolute best neutralizing antibodies. And that's important because not all of us are going to be making the best neutralizing antibodies in many cases, and so having this other thing, and at least the facile explanation is pretty simple: is if you have a neutralizing antibody, you're preventing the virus from ever getting into the cell, and so you'll never even notice that the virus has come in because it just goes away right away because of the antibodies. But once if the antibodies are not as good, you will get viruses invading cells, and then they'll start a whole cascade of replication and dispersion, and it's you start to feel sick, and that's what's going to happen. So that's where you need the T cells, because the T cells can tell which viral infected which cells are infected with the virus, and then kill those cells specifically, or there are other mechanisms to deal with them. Have the next slide. Right. So So of course, lots of people have been working hard on the T cell and antibody aspects of SARS-CoV-2. That's kind of like everyone's attention has been drawn to that. Everyone with any scientific chops that are relevant. And so one of the first movers and one of the best people in this area is Alex Setti at the La Jolla Institute of Allergy and Immunology. And he's done just terribly extensive efforts to look at T cell responses. He's identified dozens and dozens of significant T cell responses in both CD4 and CD8 types of T cells. I should explain that CD4 T cells are important for making good antibody responses because they help. They're called helper cells because they're key to making good antibodies. So that's another aspect of T cell biology that's we've known to be very important. CD8 cells are typically the cells that kill the virally affected cells. And so there are a lot of specialties in T cells, but there's also a lot of responses. And what the other thing that Alex has found very recently is that these T cell peptides that are being recognized are not mutated in the variants. You know, the variation has happened in the spike protein, and that's very important because in some cases the antibodies against that you might have gotten in a vaccine are not going to work as well against these mutants, and that's why everyone's worried about these new variants that are coming up. But he's just found that in fact the T cell responses are not affected by these variants. In some cases, the antibodies against that you might have gotten in a vaccine are not going to work as well against these mutants, and that's why everyone's worried about these new variants that are coming up. But he's just found that in fact the T cell responses are not affected by these variants. The other thing is there are a lot of T cell responses. So with if you're trying to interfere with viral entry, you've got to target the spike protein, and you just got a few choices. And I think there's also evidence that other antibodies than spike could also be effective in preventing infection, but that you don't have a lot of choices there, and that's where the variants come in and mess things up. But because the T cell responses are very broadly distributed, are much harder to get around, as Alex has shown. Next slide, please. Right. The other way you can look at what's going on in terms of T cells versus B cells, antibodies versus T cell effects, is to look at people with antibody deficiencies. And it turns out that there are lots of people who are deficient in making antibodies. And they are, of course, more vulnerable to infectious diseases, including COVID-19. But what's interesting is this little paper that came out also at the end of, the middle of last year, then uh, and from Italy, I believe. And what they found is interesting is they looked at a whole bunch of people with antibody deficiencies, and these are some of them there. And CVID is called common variable immune deficiency. Crappy antibodies, XLA, crappy antibodies, or really almost no antibodies, and yet these people are not mostly dying. There's one death. There's 3 ICUs. It doesn't look like these. I mean, they're vulnerable. They're at risk, but in some cases they don't even have any symptoms. In this one example, so and high fever. So I had to. I'm not a real doctor, so I had to look up what dyspnea is, which is shortness of breath. So now I've learned another useless medical jargon term. Shortness of breath is this thing. So that's great. So now you learned some medicine too. I have. It doesn't seem like this is devastating. That lacking antibodies is not a good thing, but it's not devastating. And we can't say, that T cells are the plan B here, but they probably are, because again, there's only 2 highly specific mechanisms in the immune system, and it's the B cells making antibodies and T cells making T cell receptors. Everything else is much more sort of generic, and not specific. So of course, as a T cell person, I'm pretty sure that there must be T cells. Next slide, please. Right. This is a highly simplified, crappy picture of T cells. There are many flavors of T cells, and this is very, yeah, uh, many different types of T cells. I told you about the helpers versus the killers, but there's actually quite a number of other distinctions. Have the next slide. Right. So I just want to give you a little bit of flavor of some recent work that we've done that isn't published yet, but we hope it will be very soon. And this revolves around a kind of labeling method that we've developed. We had a very popular one years ago that multimerized peptide MHC complexes, and turns out that they could bind very cleanly to specific T cells based on the multivalent interactions. Recently, a postdoc in my lab, Vamsi Maladjosla, has done one better and developed a whole new platform for labeling T cells. And he calls peptide MHC Spheremer. And this has 12 peptides. Those who are keeping score at home, this has 12 peptide MHCs displayed and binds T cells much better. We get more T cells of a particular type. We then test, are they really this type? And they are. They have that specificity. How we were, well, this is just showing some of the data, and for most of you don't know or care, I think, what these things are, but the red, if you look at the red there, it shows that it's better. If you look at binding characteristic, it shows that it's better. It's also cleaner. There's better signal to noise at capturing specific T cells. Have the next slide. Yeah. So here's the heart of it, is that it turns out, and you can see over on the left, there are 4 other coronaviruses that have been circulating for a long time in the world. They have names, but more importantly, they have, in some cases, a lot of similarity in their sequences to SARS-CoV-2. And it's been speculated, and there's been some data from Alex Setti and Shane Crotty to say that they could see some in people that had not been infected with SARS-CoV-2, they could see some T cells coming up to these conserved regions, but they couldn't say too much about them because of the limitations of their assay. So Vamsi looked carefully and compared across the whole SARS-CoV-2 genome, where were these most conserved sequences, and turns out there's this open reading frame 1AB, has a colorful name, is actually one of the more conserved genes in the virus, more than spike. So S is spike here in the middle, and N is less conserved, as Sean will mention. But these and red is a high similarity, and there are gradations. There's on the lower right, there's a bar there, and we've confirmed that these are cross-reactive between the different strains, some of the ones here. And the next slide. And what convinces us that this is significant is with this staining reagent, we're able to look at the phenotype of the T cell, phenotype being that what are the characteristics, what kind of T cell this is. And we know a lot about the different kinds of T cells. And but the only kinds you have to know about here are what's called naive and memory. And naive are T cells that have never seen this particular antigen. Memory cells are what happens if this is a recurrence of something that the immune system saw earlier. And these are the cells you want. Memory cells are tremendously protective. and they're ready to go. They are primed to get going right away. Naive cells take time to reorient themselves, and if you have that time, that's great. But if it's a fast-moving virus, you don't have that time, and that's where you want memory cells. And that's part of what vaccination is geared towards, is to create uh, a pool of both the T and B cell memory phenotype cells. Um, and so we were very pleased when we looked at people. These are people now completely naive. They've never been exposed to SARS-CoV-2. In fact, samples are collected typically before the pandemic. And what we can see here is, if you look in the middle here, I wish I had a pointer, but I guess I don't. Uh, for the P1 peptide, it's a little, sorry, it's a little obscure. So P1 peptide are unique to SARS-CoV-2. But now then you go down to the lower left and look at P5, you see something very different, which is you see more T cells staining with this peptide reagent, and they're all memory, basically. The upper and lower left panels, sorry, this isn't labeled this well as it should be, um, are the memory cells. And you see highly skewed towards the memory cells. That says that these T cells saw something similar to P5 once upon a time. Similarly, the P10 peptide and the P17, again, we selected these for sequence homology and cross-reactivity, and we're seeing they have predominantly memory phenotype. Whereas up in the upper right side, P1, P19 are very unique to SARS-CoV-2, and you have naive cells. So you don't have any prior experience of that. So this fits very well with a scenario whereby many people will have memory cells for these conserved peptides, particularly in the open reading frame, but also some are in the spike region. Of the next slide, please. And this has what you might expect, given what I told you about how valuable memory cells are for a given specificity. We find that these conserved T cell specificities are both uh, are elevated, prominent in the responses of SARS-CoV-2 patients. And this has been a great collaboration with Kari Nadeau here at Stanford, who's been just amazing at getting samples from COVID-19 patients and so forth and so on. But what you can see here in purple are the people with the mild disease. And what you can see is that for P5, P10, P17 is on the other side. Show these are very prominent, and these are people that had a relatively mild form of the disease. Whereas the people that had a more severe case, which is the orangey one, invariably were making responses to the most unique parts of the virus, and not to these. That's a good thing. Uh, so I think that's it. Next slide, please. Right. Well, we have conclusions. So I think we're hope I've convinced you that T cells are an important element in immune defense. They are important in vaccinology. It's just that the way you can analyze T cells is technically much more difficult. It's taken a lot of effort over years and years to start to develop some of the tools that you're seeing here and others that I'm not presenting, to give us a much better idea of what the T cells are doing. And then to see whether that makes a difference. Can you see differences in T cell responses that correlate with good outcomes versus bad outcomes? And that's what I think you tried to show you a hint of what that could be. So with that, I think I will go to the credits and in the last slide, and just thank all the people that contributed to the work I showed, particularly Vince Melody Drusilla, who has been phenomenal in developing this new technology and doing largely doing the experiments I showed with help from Connor Ganjavi, Alana McSween, Julie, Anna Jimena, Allison, and Karina Do and Manali in her lab, Taya Wong and Saborna in her lab, and the funding from the Gates Foundation and Howard Hughes Medical Institute. So thank you, and I'll stop there and turn it back to Andre. Is that?
Yes, thank you, Mark, very much for the presentation. And now I'll turn it over to our own Dr. Sean Tucker.
Obviously, Mark's really hard to follow. I'll do my best, but let me tell you a little bit about our approach. Obviously, we're in the business of making vaccines, and our desire was to make a vaccine platform where we could hand out something to be very simple, like tablets, because they're easy to administer, easy to distribute, and of course, we just confirmed the obvious last week. We did a survey and found that hey, people really prefer to have a tablet rather than injection. So, I mean, again, a survey says, and everybody you ask will say, give me the tablet. So the key thing from our standpoint too was we wanted to build a platform technology that we could use over and over again and not have to start from scratch. And we developed such a system for oral. And again, you probably are aware that there's definitely gene-based vaccine systems out there that work for any antigen that's injected. We would be the first from the standpoint of having a oral technology where you could essentially cassette in your gene for norovirus or gene for coronavirus, put it into our platform, and you're off and running, and you could create an immune response that would be predictive to be protective. And next slide. So that is what we developed, a platform. And when I say, again, a lot of people have the desire to build what has been called the Holy Grail, an oral vaccine platform. We actually have data that says actually we can make, essentially a platform that generates immune responses against whatever genes we've set into the vector. And whether that's COVID or coronavirus or norovirus, or, we've been able to do this. The key thing I want to mention here, and Andre alluded to this in his discussion, is the fact that when we did a study in a head-to-head comparison between our vaccine and the market-leading injected vaccine, as you expected, neutralizing antibody responses were really critical for the success of that vaccine in terms of and predicted protection. For our vaccine, it wasn't as important, and we had like 1/10 throughout the essentially the serum antibodies levels that vaccine did, or even more 1/6. But really, what it came down to is we actually did just as well in terms of protecting against infection and certainly infect and illness. And I think that's a really important thing. And what we found with our best correlate of protection in that study was the cellular response, and a cellular meaning making a B cell of IgA class that recognized HA, but it didn't have to be neutralizing, but that seemed to be the most important correlate. And there was also other T cell correlates of that as well. And again, that suggests that our mechanism of protection may be much different than what happens with the injected vaccines. We're really making that IgG response that blocks the interaction between virus and cell is absolutely critical. Next slide, actually COVID. It is a significant health problem, and we know the vaccines are highly effective. And as Mark said before, neutralizing antibodies against the match really works well, from the injected vaccines. But there are certainly lots of questions and concerns, and I'll just give you a couple of mine. First, one of my key questions would be is we don't really know how long immunity will last for the SARS-CoV-2 and the vaccines. And we know from the common cold coronaviruses that are out there, and Mark talked about it, 229E and a couple others, what we do know is the natural immunity lasts 2 to 5 years before you can get infected again. And it doesn't appear from those studies that you actually are losing antibody titers, which is causing you to be susceptible, but that the viruses just mutate. And we do know that RNA viruses have that ability to mutate. And this is, again, something it's just a question is how long these vaccines are going to work. So the next thing is one of the big concerns. Obviously, if you have one virus, that is in circulation, it's a lot easier to generate an immune response that's going to be protective. But SARS-CoV-2 is not a single virus, and it's a family-related viruses. And right now, it seems that there are mutants that are showing up that are essentially able to circumvent the neutralizing antibody response. And as Mark said before, it's better to have sort of a backup plan in that case, or something that could essentially be cross-reactive. We know that, for example, the South African strain, the vaccines don't protect as well against that as the potential strain. And what the vaccine manufacturers are now doing is they're deciding to pursue match strains. And this could be an effective strategy. Certainly you make a new vaccine every time there's a new strain that pops up. And if there's a very limited number of viruses that can circumvent this, or you can make, that seems effective. Or if you have a way to basically generate herd immunity that's quick, because let's face it, it's not making vaccine that's really important from the standpoint of protection. It's getting enough herd immunity out there so that you could suppress, in a global situation, the viral outbreaks and be able to return things to normal. So I just think that's really important to keep in mind. It's really herd immunity is what you want, not to make a vaccine. So I would argue, that as vaccine manufacturers improve their capabilities, the rate limiting step is really not the time that it takes to manufacture, but the time to administer the vaccine, because it just takes a long time to inject, hundreds of millions and billions of people. And obviously, in the middle of this slide, it's just there's severe limitations for the rollout of a needle solution during a pandemic. And I won't go into the details, but it's just difficult. And that's what we know. And again, the key thing is that the absolute goal is to make long-term immunity or herd immunity, not to make vaccine. So again, I'm just going to reiterate my point. The current pharma approach is we're now heading toward is a cycle of discovery of new strains, make a new vaccine, manufacture a new vaccine, administer the new vaccines, and do this in a very global scale. And if variants are appearing faster than the vaccines can be administered, we're going to feel like hamsters on an exercise wheel, and it's going to be really hard to achieve that real thing where it's, herd immunity. And the question I'm going to ask now is, can we get off that wheel by taking a different approach? And I'm going to tell you a couple ways that the Vaxart approach may be important from the standpoint of getting off the hamster wheel. Next slide. Back to my analogy of the hamster wheel, we're basically chasing the vaccine if the approach is basically to continuously make strain match vaccines. And there's two ways that I think we could get off it. One, we could provide tablets. It's a lot faster to hand out tablets or send them by mail than it is to inject, 100 million, 300 million people. That's pretty clear. And you could get out to herd immunity a lot faster if that's the approach. The other approach that we've taken, and this relies on basically our understanding of our platform, the way it works. We believe that IgA can be much more cross-reactive than IgG. It's a big, dimeric molecule or polymeric molecule that can go and glob onto viruses and doesn't necessarily have to really bind in the receptor binding domain, and that could be important. And the other thing is to really target T cell responses. And as Mark suggested, there's definitely evidence out there that T cells can be very important. We, back in March of last year, were very concerned about the S protein and the fact that it didn't seem to be as conserved to us as the N protein. And so we included not just the S protein in our construct, but the N protein as well. And the reason, again, was that we thought that N was more conserved. In the variants, for example, the South African strain, I believe there's like 6 to 20 mutations in the S protein. There's only one mutation in the N protein. It's much more conserved. So that was why we took that forward and went to the clinic. And with that, next slide, let me tell you about this approach was mostly single dose, what we considered low and medium doses of our vaccine, and only a small cohort was boosted. We believe that we've met the primary and safety efficacy or primary endpoint safety and immunogenicity endpoint for this study. Next slide. In terms of adverse events, obviously that is the primary endpoint for this study. Let me just go through this. Essentially, there really isn't much to be concerned about. There was no severe adverse events. There's no adverse events of special interest. We're not seeing the same sort of incidence of headaches and fever, and certainly not injection site pain that's been reported for the competing COVID vaccine approaches. There may be a slight uptick in, some gastrointestinal, but there's nothing to be concerned with. All these things seem to be mostly mild. Next slide. Now, we didn't see neutralizing antibodies of the serum, but we did see a very strong T cell response. And Mark talked about, how difficult it can be to measure these things. And essentially how we did this is we took cells from people that participated in our trial, used peptides. Again, Mark talked about the importance of peptides in terms of what T cells recognize. We took peptide libraries, basically they're chopped up pieces of either the S, and just measured the immune responses and, what types of cytokines are made. And certainly what we found is a very Th1 bias response, which is good. We're making more gamma and TNF rather than IL-4 or IL-13. And again, well, as expected, we're getting responses not just against the S, but we're getting it against the N as well. And in fact, what we thought was really amazing was we were getting some really, really high responses against the S. If we go to the next slide, let me tell you, a little bit more about the details. Again, showing the subjects, at day 1 versus day 8. As I said before, we're taking cells from people and basically stimulating with the peptides, against the S protein, and just measuring the immune response by measuring gamma interferon intracellularly, and you can, or TNF or CD107, and we can see a very much an increase in that response. And it seems to be very, and it's very antigen specific. If you give peptides, for example, to flu, a control peptide against flu, basically you don't get the same sort of like increase at between day 1 and day 8. And these responses are, considered from our standpoint really high. We're getting subjects that are basically making gamma interferon in more than like 1.5% on average of their CD8 responses, and CD8s are the cytotoxic T cells, the ones that can go out and basically hunt down and kill virusly infected cells. And because these numbers seem to be higher than's been reported in the literature for other people, as well as because we hadn't seen anything like this from our own studies in the past, we wanted to do something to really sort of make sure that we were on the right path, that our analysis was basically giving us something interesting. So I'm going to go to the next slide and tell you about a little study we just did, which is where we basically, this is not a head-to-head study, but what we did is we took cells from people that basically voluntarily got the either the Pfizer or Moderna vaccines, and then ran in parallel in the same assays, the same time points, samples from our clinical trial, in a comparison. Again, it's not a head-to-head comparison because we weren't able to use the Pfizer Moderna vaccines within the same clinical trial. But again, we're taking the same cells, same assays, and basically just measuring, whether you get gamma interferon or TNF, on the CD8 population. And what you can see is it really did quite well, the Vaxart vaccine. We're getting basically the same subjects had a pretty equivalent response that we saw before, and the Pfizer and Moderna are getting responses that are much more in line what has been reported in the literature. So again, from our standpoint, it looks like we compare quite favorably on a T cell response against SARS-CoV-2 responses and how you make gamma interferon and TNF. If we go to the next slide, one of the things, again, is that we wanted to know whether, the vaccines could basically be cross-reactive against the variants. And as Mark had said earlier, essentially, and this is a study by, again, the SETI lab, or this is Alison Tark, and another paper of hers. She seems to be very prolific, but basically looked to see how the RNA vaccines did when they compared against the strain match peptides, as well as the ones that are from South Africa, UK, Brazil, or California. And what they found, although the numbers seem to be, reasonably low in what we've seen in the past, they seem to be consistent. So essentially, you're getting an immune response that's quite similar, whether it's to each one of these things, to the S protein. And, as Mark suggested, T cell epitopes are much better conserved and basically, are not being mutated as frequently, or essentially we're not being, right now on the T cell side of things, it doesn't seem to be circulating. So the next question we wanted to ask, well, if, the vaccine with the S is, certainly seem to be cross-reactive, at least these small strain variants, what happens if you go further? I mean, how do you basically predict in the future whether, if a bat comes and bites someone and, that person could become patient zero? How we know that our vaccine could potentially be protective or at least cross-reactive against those coronaviruses? So on the next slide, let me tell you about what we did. We took the same cells from our subjects and essentially stimulated, rather than the SARS-CoV-2 peptide library, but the S and N proteins from a number of ones. And again, they're not very identical, so this is a very hard test. And what we found is we're getting very strong T cell responses. Again, this is something on the order of 0.6% or above, or 0.4, depending on average, for these responses. And these are basically at the same order of magnitude or higher than has been reported for the RNA vaccines, or what we've seen. So this looks like we're making a very, very strong cross-reactive T cell response against things that are pretty diverse. And again, in these assays that we're comparing. So this is a very nice result for us. And, and we think there's some value to this. It tells you a little bit about our oral vaccine platform. On the next slide, as I alluded to, before, our essentially T cells are hard to study in humans, and it's hard to really do it in a definitive manner. But what's the evidence really that T cells are pretty important in humans? First, we know that cross-projective and long-lasting. When N protein responses, SARS-CoV-2 can last a pretty long time and seem to cross-react, which is great. We know that people with, as Mark talked about this Kininti paper before, people that actually have no B cells and no antibodies don't die of COVID, and they actually do okay. The authors actually made a big deal about basically suggesting that people with the the common immune deficiency, the CVID, were much worse because they have B cells, but they just seem to be defective. And when I did a little literature search, about, so it also appears that some of these CVID may have T cell responses. That, of course, that was with the academic journal called Wikipedia, but still, it does seem that these CVID patients basically are a little bit more less characterized. But essentially, as Mark's point, T cells can play a really important role for protection. We do know that mucosal T cells may be helpful in terms of blocking shedding and transmission, as well as mucosal antibodies. And we do think, and in a prospective study that was done in England by Wiley, essentially we looked at first responders, healthcare workers, and firefighters and police officers, and just measured T cell responses and antibody pre-existing, just watched them over time. And as they got infected, they just figured out that it seemed that the protection seemed to correlate better with this COVID disease. T cell responses seem to protect better than the antibodies that they were measuring. Keep in mind, this is before the advent of vaccines or substantial herd immunity. And lastly, as Mark talked about, there's definitely a broader antigenic repertoire for the T cells versus the antibody. And we do know the viruses seem to be mutating the parts that are very important for antibody recognition. With that, let me talk about one last data slide on the next slide, please. One of the things we know is that our vaccine does pretty well for making IgA responses, and this is from IgA responses in the nose. And we've already reported that we are getting antibody responses to SARS-CoV-2. But what was very interesting is that in the subjects that actually had antibody responses to matched SARS-CoV-2, those subjects also seem to have cross-reaction to all the other SARS, or all the coronaviruses that we were testing, include SARS-CoV-1, MERS, and a lot of these things that Mark talked about as well, such as NL63 and 229E. So again, what's happening is our vaccine seems to be deriving things differently. We seem to be getting a mucosal IgA response, and it seems to be cross-reactive, and we're getting a very, very potent T cell responses, particularly in CD8s, and those seem to be very cross-reactive as well. And that seems to be the data to date for our vaccine candidate. Next slide. So where are we in this thing? Obviously, we are expected to start a dose ranging study relatively shortly. We think it's around mid-year now. We believe we are going to continue into, if that looks good, we are going to continue on a phase 2 efficacy study later this year. And as Andre alluded to as well, we are a platform company. We are looking at other approaches. We have made S-only versions of things that are strain matched, and we're going to answer questions and research basically whether the strain match is much more potent from the standpoint of protection as the ones that we have that's just cross-reactive. And again, these are research questions, and then we'll advance candidates that we think are important to evaluate in humans. And with that, I'm going to turn things back to Tara to lead the question. Oh, one last slide just to thank all the actual people that worked on the materials, and particularly Susan Johnson, Calissa Martinez did a lot of the work on the T cell work, but there's a lot of people participating in our studies. So, and with that, I'll turn it back over to Sarah.
Thanks, Sean. So at this time, we're going to begin our question and answer session. As a reminder to the audience, you may submit your questions through the Q&A tab.
It would definitely, following what T cell responses and specificities are characteristic of people who do well in a disease, I think that could be the best way forward in terms of going from small studies where you're getting this kind of data to positioning yourself for efficacy. And of course, efficacy at the end of the day will be will tell the tale. But what we're looking right now for are surrogates, and I think focusing on the T cell responses of people that do well is good. I mean, we're also doing that in TB with Tom Scriba's group in South Africa. We're focusing on the T cell specificities that are characteristic of people who control TB versus the people that progress to TB disease. So I think we're approaching the dawn of a new era in being able to at least intelligently pick antigens for the T cell responses that have the potential to do the most good.
So just to answer your questions, there's the two questions first on the dose ranging. The goal for the dose ranging study is to really figure out if we can push up the antibody responses or see the dose responses. We saw in that small study, we did see that, we saw that lower doses didn't do as well for making an IgA mucosal homing B cell as they did in the low dose. The T cell stuff seemed to be pretty independent. We got pretty good T cell responses at the low dose and in the high dose. So the dose ranging study is going to teach us more about that. In terms of, asking about the correlates, I'm going to point to something, we've actually published on. In the flu study, we did this really massive challenge study. We took all sorts of samples. We did analysis. We published on, basically, what the best correlates of protection from that flu challenge study. But the other thing we did is we took some, samples that were under fixed whole blood and ran CyTOF analysis. And there's a paper that has been submitted that basically will describe some of the cellular responses that you could measure. And obviously, as Mark said, it's really hard to basically do PBMC processing, phosphopeptides, learn about T cells. But if you could find some essentially surrogates or something that's correlative that's on a fixed whole blood basis, I think you could come up with something as well, maybe not as simple as antibody, but it would be much better from the standpoint of predicting and ease of, because we hate to make the clinical people basically take PBMCs for, 300, 400 subjects.
Right. No, I'm with you. So maybe my final question, and apologies for the background noise. On the production, maybe Sean or Andre, if you can comment on what might be sort of the end of year goal in terms of, now it's encouraging you're getting some production in-house. So just curious, like if you have maybe for your lead candidate a goal for end of the year, and maybe for your the S spike focused backup kind of candidates, a certain number of doses that you may want to get to end of the year. Any qualitative color would be helpful.
Yeah,
Andre.
Yeah, Mayank, let me try to first complement what Sean said on our clinical trials. So don't think about our S-only constructs as being backup constructs. We don't think about them like that. We think that they could be potentially complementary. So we expect, or rather Sean's group expects, that the S-only constructs, you're going to see higher antibody responses and lower T-cell responses, right? And the SN construct, I think people don't fully appreciate that, was designed to be cross-reactive. We showed that the T-cell responses are very high. We are making IgA responses. We're going to antibody responses in the mucosa. We're going to characterize those. So the plan is to continue to study this portfolio. We have a lot of preclinical studies going on, and then also study in the clinic, and then think about clinical trials that are relevant in various jurisdictions, right? So here in the US, in the UK, we're going to be looking at boosting studies, vaccinating those that have been previously infected or previously vaccinated by needle, and see what kind of responses we get. we are also thinking about studies against mismatched strains, right? To show that our constructs are cross-reactive. So think about using our SNN construct that includes the Wuhan S in a region of the world where the predominant strain is South Africa, right? So we are thinking about all these combinations. And then as we discussed, the world is moving forward. I think the UK has shown signs of really moving forward with the a challenge study. And if that's the case, that would be very interesting for us. As far as manufacturing capacity, we sort of telegraphed that we have had some of the same issues that have been broadly reported by some of our larger competitors. But we have worked, the good thing is that we have worked for many months now to bring in manufacturing capacity in-house, also with an eye of being able to produce a number of vaccine constructs, not just one. And I think we are well on our way to do that this year. And we're also exploring adding other manufacturing partners that could be more strategic partners of ours, both here in the US and internationally, also because we would like to decouple the progress we make in COVID with the progress we're going to make in norovirus. For us, these are the two significant value drivers this year. And these two programs are very different because COVID, as you appreciate, is a vaccine program that all around the world kind of depends on governments, right? They are your end customer. And it's also a program against a virus that still evolves as far as of our understanding of it. Whereas norovirus is entirely within our control. So we'd like to be able to decouple the two so that delay in one doesn't cause a delay in the other. But I'm not going to give you specifics now. It's just that we feel comfortable as far as the progress we are making right now on the manufacturing front.
Thank you, Andre. And thanks, Mayank, for the questions.
Quickly, I'm going to fire off some questions before we run out of time. First one, how important is T-cell response in providing variant coverage? Do you think a stronger T-cell response could drive stronger protection against variants?
Well, from our standpoint, it's a lot easier. I mean, and I'll ask, Mark next. It's obviously easier to escape an antibody response than is a T cell. And as Mark said, at a minimum, it's a great backup plan. It's also possible that if you're talking about trying to address not just one virus, but a family of viruses, obviously T cells could play an incredibly important role from the standpoint of providing a protection. I'll just ask Mark if he has any additional, color or commentary.
Yeah, no, I completely agree. That's it's just that we have not had good metrics for correlates of protection for T cells. We have those for antibodies. We had them for many years. But the failure of many vaccines has taught us that there are things we don't know about metrics of vaccine efficacy until you do huge trials and spend a lot of money, like we have in many cases for HIV and other vaccines. So I think we're the prudent thing is to hope for a really good T cell response. And I think I mentioned some of the tools we've used. Sean has mentioned tools that he's used to take a look at that and then pick what looks best. But you want T cell responses. Antibodies are not enough.
Great. Thank you. Next question, how does T cell response differ in adults versus kids?
That seems like a Mark question to me, so.
Is that right?
Well, since I haven't done any kid studies yet, so.
Well, no, I think that was something I wish I had said in my presentation is, there's a central mystery about why are kids so relatively immune to SARS-CoV-2, which is not true for measles, is not true for many other infectious disease. Kids are actually more vulnerable. So part of what I showed is how memory cells for these circulating coronaviruses are a good thing in terms of a milder form of disease. And depending on what reports, but I have reports to say that these other coronaviruses are rampant amongst young children. And so my guess is that they have spectacular, we're looking at, actually this week, we're looking at some samples that of children, and we hoping to see that they have really good responses, but we don't yet. So, but that's the implication. These diseases, communicable diseases like this spread lots. Anyone who's put a kid in daycare knows they come home with all kinds of diseases because they're slobbering all over each other, and so forth and so on. So and then it also makes sense that older people who are not in daycare anymore, and the responses fade, and we know elderly people, especially, all the responses tend to fade, and that's why they're more vulnerable.
Got it. Thank you. And then one last question from Yasmin Rahami from Piper. Asks, do you think stimulation of mucosal immunity via oral or inhalable formulation would have similar immunogenicity profiles?
So in our data from animals, we see a similar profile, and we published on this in cotton rats that we were getting a pretty similar IgA response, in the bowel, in the lungs, in the nasal. So I think that they can be equivalent. And we obviously know that we made human antibody responses, or we made antibody responses in humans with our oral tablet vaccine. And so I would gather that ours, I have no data that says ours is any worse. And I would say that it could be just effective. The other thing too is that if you're talking about an oral adenovirus vaccine, I think that the anti-vector immune responses will make it harder on the intranasal to boost than it will on us, because I would expect their anti-vector responses to go up.
Thank you, Dr. Tucker. And Dr. Davis, I will turn it back to you, Andre, for closing remarks. Thank you.
Yes, well, thank you so much to I understand, over 1,200 people that logged into our call. I hope that you found it informative, and please stay tuned for more developments from us. Sean will be presenting tomorrow at the World Vaccine Conference, and we'll be discussing a little bit some of the antibody responses. With that, thank you very much for joining us today.